So enzyme topic is a small topic, but many questions are asked from this topic, and it is a very conceptual topic, very easy to do this topic. So the first definition, we can say, what are enzymes? Enzymes are proteins, and all enzymes are proteins, we say, but ribozyme is RNA which act as enzyme. So this is one exception here. Ribozyme is RNA acting as enzyme, right? Now, you must have played one video game called Pac-Man, in which this kind of structure has to eat the dot. This kind of structure has to eat the dot here in this video game. And in this video game, we have to finish all the dots, right? So this is Pac-Man and this is that dot. Now let's say this is open mouth of Pac-Man. So this is upper lip, this is lower lip, and here teeth in the center, right? Now I'll explain something with my hand. See here. Let's say this is open mouth of Pac-Man. This is upper lip, this is lower lip, and teeth here in the center. So how this Pac-Man will eat the dot? This Pac-Man will first hold the dot with the lips. Then it will cut the dot with the teeth, right? So now let's say this Pac-Man is enzyme and this dot is the substrate. This enzyme has to eat the substrate, right? So now see, the mouth of Pac-Man now represent active site of enzyme. Active site is further made up of binding site. The lips represent binding site and teeth represent catalytic site, right? So active site is made up of binding site and catalytic site. And now this enzyme will first hold the substrate with the binding sites, and then it will cut the substrate with the catalytic site, right? So note it down that binding site present here, and this in the center is catalytic site. So active site is made up of binding site plus catalytic site, right? Now let's say this enzyme is chymotrypsin enzyme. In case of chymotrypsin, here we have histidine, here we have aspartate, and here we have serine. So can I say, in case of chymotrypsin, which is a protein breaking enzyme, for this enzyme, which amino acid is binding to the protein substrate? That is aspartate and histidine, they are binding the substrate. But serine is responsible for cutting the substrate. So because serine is responsible for cutting, so we say that chymotrypsin is a serine protease. That is, with the help of serine, protein is cut, right? Serine protease is a very big class of enzyme. Chymotrypsin is just one example of that class, right? There are many enzymes further coming in this category, serine protease, and all the serine proteases have same catalytic triad. Of histidine, serine, aspartate. Presence of 3 amino acid at the active site is known as catalytic triad, right? So we have same catalytic triad in all the serine proteases. Now let's see the examples of serine proteases. Examples of serine proteases are chymotrypsin, we have already done. We also have trypsin and elastase in this category. Then plasmin, thrombin, complement proteins, clotting factor 10 and 11, and PSA, prostate specific antigen. These all are serine proteases, right? Recently, they have discovered that serine proteases have a role in tumor cell metastasis. That is, in the spread of the tumor, they have a role. That's why this topic is very important these days, and it can be asked, right? Now, next is, there is a term bi-bi reaction, BIBI, bi-bi reaction. This is a 2-substrate, 2-product reaction. Like we do now, one thing is oxidized, other thing is reduced. Let's say A getting oxidized. On the other hand, B is getting reduced. So there are 2 substrate and 2 products to this reaction known as bi-bi reaction. We have to do the types of bi-bi reaction. There are 3 types: ordered bi-bi reaction, random bi-bi reaction, and ping-pong mechanism of bi-bi reaction. In ordered bi-bi reaction, mostly dehydrogenases enzymes have ordered bi-bi reaction. Mostly kinases have random bi-bi reaction. And in ping-pong reaction, there are 2 examples. Serine proteases, all the serine proteases, they have ping-pong mechanism of bi-bi reaction, and also aminotransferases have ping-pong mechanism. Aminotransferase example is SGOT, SGPT, right? Now, let's say there's a chymotrypsin gene. Let's say this is chymotrypsin gene. By transcription, it will form its mRNA. On mRNA, there is a codon UCA, which is coding for serine. You know, serine is present in the enzyme or protein. So in mRNA, there will be a codon for serine. Then translation will occur and this enzyme or protein is synthesized with serine at the catalytic site, and this is known as chymotrypsin, right? Now, let's say this chymotrypsin enzyme is cutting the protein at a particular rate, at a particular velocity, right? But I want to increase this velocity. I want that the rate or velocity at which the enzyme is cutting the protein, that should be increased, right? So we will do some experiment, right? Which amino acid is responsible for cutting? Serine is responsible for cutting. So we will try to change serine with some another amino acid. So it is like an experiment which we are going to do. We are trying to change serine with some another amino acid. So let's say this is the gene, and here in this gene some mutation we have done. Mutation is done so that now after transcription a different mRNA we are getting, and now it is having CCA codon which is coding for proline. So now when translation will occur, enzyme or protein will be formed having proline at the catalytic site. So we do such experiments in order to increase the enzymatic activity. But let me tell you, in all such experiments, we are usually a failure. Because nature has already provided us with the best thing. Further, we cannot increase it, right? Even if I try doing another 18 experiments in which I will keep changing all the amino acids, I will try for another 18 amino acids, every time enzymatic activity will be decreased, or sometimes it will stop at all, but it will never increase. But here you need to know that what is this experiment known as? This experiment is known as mutagenesis, because we are creating a mutation here known as mutagenesis. But this mutation is not caused at a random site. No, it is caused at a very specific site, the site of the catalytic amino acid, right? So this is known as site-specific or site-directed mutagenesis. Site-specific or site-directed mutagenesis. A question asked was, oligonucleotide with single base change is used in. Oligonucleotide with single base change is used in this technique, site-directed mutagenesis, because in this technique, when I have given you the example, you note here that I have written initially UCA. UCA now converted to CCA. So only this first base is changed. The CA is same. So oligonucleotide with single base change is used in this technique, site-directed mutagenesis, because whenever we are doing these experiments, we'll try to change minimum, because minimum changes in gene, only minimum changes can give you better results. Like if you do more change in gene, then the results are not good, because some portions, neighboring portions of genes are also deleted, sometimes promoter deleted, so anything else deleted. So that is going to give hazardous results, right? So minimum change in gene gives better results. So whenever we do this experiment of site-directed mutagenesis, then minimum change is done. That is, mostly it is only single base is changed and amino acid gets changed, right? Your guides have given this question. When they have given this question, they write that answer to this question is RFLP, that is restriction fragment length polymorphism. But no, answer is not RFLP. See the another question. When this question is asked, single mutation is detected by, then your answer will be RFLP. This RFLP technique detail we will be doing in molecular biology, right? Now let's do some general properties of enzymes. Enzymes, they are not used up in the reaction. They will increase the rate of reaction, that is, they will speed up the reaction. Like, you know, the reactions which are occurring with the help of enzymes in body, those reactions can also occur in vitro condition. In vitro condition, that kind of reaction will take years to occur. It can occur, but it is so slow to occur, right? So enzyme will increase the speed of that reaction, and therefore enzyme will decrease the time used for the occurrence of that reaction, right? Also, enzymes decreases activation energy. They decreases the activation energy required for the reaction. Let's see what is activation energy required for a reaction. First, let's see how a normal reaction occurs. How a reaction occurs. Here on y-axis, I am taking free energy change, and time I am taking on x-axis. On y-axis, I have taken free energy change. Now, substrate, let's say substrates have this much energy. Product is having less energy. This is the product. So how a normal reaction occurs? That first substrate has to reach high energy state, only then it can be converted to product. So this much energy is first required by substrate to reach this transition state. This energy required is known as activation energy. That is, first we have to activate the substrate. Only then it can be converted to product, right? Now what enzyme will do? Enzyme will decrease this activation energy. Enzyme will decrease this activation energy. So the route followed by enzyme is this much, this one. So now the amount of energy required is only this. So this is the activation energy which is decreased with the help of enzyme. So enzyme decreases activation energy. So for earlier it was the reaction was taking time, but now when enzyme decreases activation energy, then time required by that reaction is very less now. For example, if I take the example of a mountain. So let's say this is a mountain. A person is standing on this side of the mountain and this person has to reach this side of the mountain. So first this person has to go to the top and then only this person can reach other side of the mountain. Now if enzyme is present, or if we want to decrease the time required by this person to go to other side of the mountain, what we can do is we can create a tunnel in the mountain. A tunnel can be created in the mountain, so that now this person can just go through the tunnel and reach other side of the mountain. So this kind of work is done by the enzyme, a tunnel created, and so time required by enzymatic reaction will be much less than a reaction which is occurring without the help of enzyme, right? So enzyme decreases activation energy, they decrease the time required by the reaction and they increase the velocity of the reaction. But enzyme is not doing anything to the equilibrium of the reaction, and enzyme is not changing the free energy of substrate or product. So you should also be knowing that what enzyme is not doing, right? This is it with the basics, and serine proteases also we are done. This is it.